Welcome to Edison Open House Global Healthcare 2022. In this session, we'd like to introduce you to Renaissance BioScience, a Canadian company whose bioengineering platform technologies are used to address a wide range of environmental and health problems. And with me is its CEO and co-founder, John Hughesnick. Hello.
Hello, Vivian.
Now, tell me first about your technology platform and the kind of applications that it's currently being used for.
Well, as you mentioned, we are a bioengineering company, and we primarily focus on microorganisms, primarily yeast, a very safe platform, and bacteria. And we look into also safer bacterial uses in this space. So over the last few years, we've become very adept at what we call whole organism engineering, as well as the classical standard genetic engineering, and also combining these two techniques. So much so that we've already made and licensed over a dozen products to a number of global leaders in the food and beverage space. At this time, what we're excited about is a new area that we're going into, and that is the oral delivery of RNA using these very safe delivery systems, particularly yeast, baker's yeast. And this has broad applications into agriculture, and agriculture broken down into pesticides for crop protection and for animal health or companion animals, aquaculture, and even eventually human therapeutics as well. So we're quite excited about this oral delivery of RNA in a very safe and effective manner.
Now, we all know the word RNA, or because we've come across it through COVID, but basically it's that little messenger bit that shuttles between the DNA and what things are eventually turned into. So I know that you work a lot in biopesticides. What's driving the demand for alternatives to chemical pesticides? And it's not just concern about the chemicals, is it?
No, I think there's a lot going on, especially now. Look, the global agricultural business, as we speak right now, already has a certain toll. And in some cases, environmentalists are saying the toll is already great. This is just going to escalate as our population grows. So there is an absolute need for a sustainable way to reduce some of these crop losses due to pests. And this sustainable way, we believe, is not going to be chemicals, but something new, novel, that it's going to be a different approach. And this is what RNA, this whole new area of RNA, which is very specific to the pests, much more environmentally friendly, and has a broader application with new ways of approaching these problems for this global kind of agricultural space, but in a sustainable way.
So how do RNA-based pesticides actually work, and what advantages do they have over conventional types of pesticide?
Well, again, I think the key point here is how they're going to be working in this space is, as you had mentioned, there's that classical molecular biology dogma, if you will, where it goes DNA to mRNA to proteins. And then there's a flip side to that, right? There's RNAi, RNA interference, RNA inhibition, if you will. And that is basically, instead of making a protein, you're shutting it down. You're reducing the levels of those proteins. And if you target a protein in a particular pest that's essential for life, or you can even multiplex, where you can go after multiple essential genes, if you will, all in one system, so it's not conducive, if you will, to resistance, you can actually then shut off those genes and harm that pest specifically. Again, because the RNA is based on the DNA. So it's the DNA of that organism. And hence, we kid around sometimes that we could make a very, very potent pesticide that would kill a particular insect very effectively, but you and I could actually eat it because it wouldn't affect us. It's the carrier itself would be also very safe in this case because it is a yeast, which we consume all the time as well. So that combination is something that we're quite interested in going forward. But RNA on itself, very specific. It's a nucleotide, so it's biodegradable on its own. And I think that's one of the key areas, because it is so fragile, is so biodegradable, hence lies the issue with delivery.
So no residues for a start, and presumably there isn't you don't get a case where the insects or whatever it is become resistant.
Yeah, I think that's the key thing right out of the gate is that there will be no residues. And I think that's a huge bonus in this system. In fact, even if there was off-target spraying, so you didn't hit exactly the crop that you were looking for, it went into the river, went into the soil, again, it would be harmless to all the other insects, animals, and humans as well in this case. So that's important, as you had mentioned, and I think that's going to be very important going forward.
And what's special about Renaissance's BioSciences production and delivery mechanisms? You mentioned yeast there, and of course that's a very familiar and ubiquitous organism.
Yes, exactly. So being ubiquitous, that it's in, you find it in our bread, you find it in our wine, and find it in beer, you find it in cocoa fermentations for chocolate and coffee fermentations for the manufacturing of coffee. It is everywhere. And it's also produced very effectively globally in multiple large fermenters and companies by the billions of kilograms, if you will, on a global area. So we're very accustomed to this microorganism, if you will. We've studied it very well because it has that kind of commercialization already there. So we have looked at this yeast, and we were able to actually make a patent by looking at this yeast that naturally harbors RNA already. It has double-stranded RNA viruses that it uses, and it's one of those organisms that actually doesn't have a classical RNAi components of dicer and arginine. So it harbors RNA. And if we knock out a few more genes in that pathway, if you will, in the organism, we can actually get extraordinarily high levels of RNA to be manufactured in the yeast itself. So that's the essence of our patent. Really, you can turn on genes, turn off genes naturally in the yeast. So it's still a yeast, but you just turned off natural ones. And you can get extraordinarily high levels manufactured of whatever RNA effector molecule you want. Now, the second component of this, which is really important, is that because yeast has it's a microfungi, it has a nice cell wall, and it really protects that RNA through extraordinary different environmental conditions. We can actually boil our yeast. And when we were doing stability studies and we were saying, okay, let's inactivate the yeast, the first thing we did was, let's see what temperatures where we will lose RNA. So we typically heat the yeast to 80 degrees Celsius. So 80 degrees Celsius. We even boiled the yeast to kill it, and we could still completely protect the RNA on the inside. So this would be the RNAi, double-stranded RNA. So you have different ways of killing yeast, but right now, from a minus 80 freezer to 80 degrees plus centigrade, we have complete stability. We've left things in the shelf for months, and you can pull out the RNA very effectively. We've done trials with people that have gone through feed manufacturing with high friction, high heat, and putting the yeast into that kind of manufacturing setting, that robustness, the RNA is still protected. I don't think there's any lipid nanoparticle, and correct me if I'm wrong, that could actually go through those such harsh environmental conditions and still protect the RNA the way we do with our yeast system. That's a manufacturing unit, as well as the delivery unit that can be sprayed, that can be also then used very safely. As you know, it's inactivated, what we may have not mentioned, but it is an activated yeast, so it is dead. And so that it would actually be very, very quickly consumed and eaten and would be only harmful to whatever you're targeting in the case of a biopesticide.
It's fascinating technology. And tell me very briefly about acrylamide and toast.
Oh, of course. That's one of our technologies that we noted that acrylamide is basically formed by asparagine and sugars. And if you have these two components in any kind of food, that goes above 120 degrees Celsius. So, for example, I always use the example of a potato. A potato has loads of asparagine and reducing sugars all combined, but it has no acrylamide. You can boil that potato, making mashed potatoes, as we would say here, and no acrylamide. But if you take that and you deep fry it, or you fry it at above 120 degrees Celsius, or even on the stovetop, if you will, that's when acrylamide starts to form. And acrylamide is asparagine and reducing sugars combining in high heat. Acrylamide is a carcinogen. So what we have is a yeast that's naturally present and found in the environment. And we screened a biodiversity of different Saccharomyces cerevisiae strains that normal baker's yeast, and we found some that have a cell wall associated asparaginase. So the cell wall associated asparaginase is already there, but it's turned off most of the time when you're making bread or you're making biscuits or wherever this may occur. So basically through adaptive evolution, laboratory evolution, and it's all non-GMO, we were able to train the yeast or ask it very nicely to make sure your asparaginase on the cell wall is on all the time. So it's literally a live yeast, and in its cell wall and periplasmic space as well, it's full of the enzyme asparaginase. And this asparaginase, when you put the yeast carrying this asparaginase in any kind of food, you reduce acrylamide formation prior to it being formed.
Fantastic. It's great. It's great. It's great stuff. Very quickly, if you would, I know that you've had some funding from the National Research Council of Canada. Is that sufficient to take your technology for use in biocontrol applications through to commercialization? Just a very brief answer.
No, it is not. The funding that we did receive, that non-dilutive funding, is really for our platform development. So we developed the platform. We're already working with other government agencies for further non-dilutive funding to actually take the next steps to commercialization to help in those steps as well. But we always like non-dilutive funding, and we have a great reputation with the government funding agencies here in Canada because of the successes that we've had, even on the commercialization front.
So just to conclude, what events should investors look out for in the upcoming 12 months?
Well, the most important thing that we're kind of looking at right now is we're doing field trials. We had some great results on a pest called Colorado potato beetle that we did last summer. Now, one of these insects, it's very difficult and almost impossible to rear.
The big stripy thing, isn't it?
Yes, exactly. And we're doing field trials this June and July. We're really looking forward to that. So we're going through manufacturing the yeast and all those steps. And then we've got all the permits and everything else lined up for that. And we'll be doing that in June and July. And at the same time, we're opening up other fronts in aquaculture, as well as mammalian models that we're working on, which would be more equivalent to that animal agriculture and human therapeutic side. So there's licensing opportunities that we are pursuing, and we hope to have closed by the end of the year. But primarily those field trials will really show our technology off in the field, where it's the sun, the rain, all those things being tested all at the same time.
It's the future. Thank you so much for talking to us. Very exciting year coming up for you. Thank you.
Thank you.